Today I'm here at the EBMT, and we reported our findings in mouse models, which we are trying to extrapolate to the human situation. We found that not only the T cells are the major players in acute graft-versus-host disease, but also recipient-derived myeloid cells, in particular neutrophil granulocytes, which we find in large amounts in the intestinal tissue. And we found that those cells provide signals for the incoming donor T cells, as well as direct tissue target via the release of reactive oxygen species that cause intestinal barrier damage, leading then to the translocation of bacteria and pathogen-associated molecular patterns. So in our work published in 2014 in Nature Medicine, we were the first to show that those neutrophils are attracted towards the intestinal tract via the release of pathogen-associated molecular patterns. We studied that by using neutrophils deficient for different toll-like receptors that would not recognize certain pathogen-associated molecular patterns. We could show that in this situation the tissue damage was significantly reduced. And this tissue damage is the early trigger for acute graft-versus-host disease that then later leads to the disease that we know in patients, and that manifests clinically by tissue destruction of the liver, the intestinal tract, and the skin. And by a better understanding of the pathomechanism of graft-versus-host disease, we were able to contribute to the development of novel therapeutic strategies. As many of those innate immune cells express receptors that signal via the JAK-STAT pathway, we studied antigen-presenting cells, in particular dendritic cells, with respect to the role of JAK-STAT signaling by using specific inhibitors. And we found that those inhibitors would decrease the activation status of the dendritic cells, thereby reducing graft-versus-host disease, in particular via the expression of MHC class II molecules. So MHC class II molecules are important as the antigen-presenting cells prime donor T cells that then later cause the disease. This all has been developed in the mouse model, but meanwhile we have already collected retrospective clinical data from patients with steroid-refractory graft-versus-host disease, which were treated after steroid failure with JAK inhibitors, in particular ruxolitinib. And we found high response rates, which motivated us to continue with a prospective trial. And now there's a phase 3 prospective trial ongoing with ruxolitinib in the context of acute graft-versus-host disease that has become steroid refractory. It's a multi-center trial involving multiple countries in Europe, Australia, Asia. And we are curious to see what if our findings in the mouse model and in the first patients can be confirmed in such a prospective trial.